to Bio-Explaining. I hope your preparation is going on amazingly. So we've come up with another practice video where I'm going to discuss various diagram-based questions on your ICSE Class 10 Biology syllabus. Stay tuned with us and practice till the end. Let's start checking. So this diagram that is being shown to you here, that represents a cell cycle. I guess all of you have understood that. So let's see what are the different parts. First, let's start with A. A shows interphase, right? That is, interphase is the time. It is also known as the resting phase, during which your cell prepares to divide. Okay. Now, within A, you see we have three divisions, B, C, D. So B shows the G1 growth 1 phase. So this is the phase wherein the cell will increase in size and it will synthesize the materials that will be required in the subsequent phases. Okay. Then you have C, which is your S or synthesis phase. S or synthesis phase means that in this phase, duplication of the DNA content occurs. The chromosomes will divide, but they remain attached. That means in the synthesis phase, if you had chromosomes like this, now the chromosomes will be like this. They divide, but they're still attached. Okay, so chromosome number is not doubling. Chromosome number is the same. The DNA content has doubled. Okay, synthesis of DNA. DNA content gets doubled. Then you have D. D is the G2, that is growth 2 phase. So here further the cell increases in size and it is now about to enter into the next phase. What is that? That is, E shows your prophase, F shows the metaphase. We have studied in order, right? Prophase, metaphase, then anaphase, and then we have the telophase. So E, F, G, H, prophase, metaphase, anaphase, telophase. All of this together, prophase, metaphase, anaphase, telophase, gives your I here. What is that I? It is known as the M phase, mitosis you can say. Or since this is the division of the nucleus, you can also call it to be karyokinesis. Okay, why do we say karyokinesis? The meaning is the nuclear division. Okay. And then what's left? There we have J left. So J is the cytokinesis. That is, once the nucleus has divided, now the cytoplasm will also divide. That is, at the end of I, you have a situation like this. Two nuclei, two daughter nuclei within the same cytoplasm more or less. Right? And when you come to J, when J is finished, you now have two small daughter nuclei. Each of them will start their journey into their own cell cycle. Now again from G1, S, G2 and so on and so forth. Okay, so I guess this is very clear. Okay, now what? So this is the diagram of a root hair, or you can specifically say root hair cells. Okay. What is part 1 showing? This is from where does a root hair come out? So we know root hairs have their origin in the epidermal cells. So 1 is epidermal cell. 2 is the cell wall. See, it's pointing out to the outermost layer, the cell wall. C is showing you just the inner layer, that is the cell membrane. So 3 is cell membrane. 4 refers to the empty space that you have. So it is the cytoplasm. And 5 is the vacuole. This is 5. The, fluid filled cavities that are present. What is 6 showing you? This part, it is the nucleus of a root hair cell. And number 7 is the root hair cell which has now completely grown. So you can call it to be the fully grown root hair cell. Okay, is it fine? The 7 is a fully grown root hair cell. And these small ones are the root hairs beginning to grow out. That is, they are still in their developing stage. They have not yet completely developed. They are not fully grown. Right? So I guess we are clear with this. Okay, identify solutions 1, 2, 3 where the cells are kept in. Number 1, you can see that the cells have all, they have shrunk. Shrinkage has happened. So that means the cell is kept in the hypertonic solution. Number 2, the cell size is as it is. So it is an isotonic solution. Number 3, the cells have increased in size to such an extent that they went turgid and ultimately burst out. The cells have bursted out and the inner components are coming out because they increased so much inside it couldn't contain. So this 3 is the hypotonic solution. Fine. Hypertonic solution. What happens in hyper? The hypertonic solution has ability that water molecule goes out of the cell, right? Water H2O will go out of the cell, right? Why? Because the hypertonic solution has less water as compared to the inside of the cell. Isotonic means it is equal concentration, and in hypotonic what will happen? The water molecules are going to enter into the cell and their size will increase. Okay? Why is this? This is because the outside solution, that is hypotonic solution, has more amount of water molecules than inside the cell. So I always tell my students ki remember using a small trick. I'll just come to that. First, let's identify the processes. Number 4, you see water molecules going out. So this is your X osmosis. Number 5, it is going out as well as coming in. So ultimately no net movement. So we say there is no net movement of water molecules. And number 6, water molecules are entering. They are coming into the cell. So this is your endoosmosis. Okay. So if you can carefully note, hypertonic solution में हो रहा है X osmosis. So I always ask my students कि remember in this way कि if X osmosis is there, who is your X? A person who is no longer there in your life, out, right? X is out. That means water molecules are going out. And after X has gone out, now how are you? You're very hyperactive, hypertension, right? Not at all at peace. So this is your hypertonic solution. Okay. If you can remember this one thing, the other one is the opposite. You all confused, no? Hypertonic, hypotonic. So X osmosis means water molecules going out and your situation is your hyper. So the cell is kept in a hypertonic solution. I guess this is clear. Okay, so a question from human physiology in between your plant science questions. Identify the diagram below. Simple. This is your excretory system. And when we say excretory system, we mean to say this is the urinary system specifically. What is part 1? It's your aorta. What's the job of the aorta? It will carry oxygenated blood from the heart to all other body parts except the lungs. So oxygenated blood from the heart, that is the left ventricle. From there, it comes to the different body parts. Number 2, what is there? Number 2 is simple. That's your kidney, the right kidney. What is its job? We all know the job of the kidney is to filter the blood, right? It takes away the waste materials. So we say it removes wastes. Okay. Then number 3 is the renal artery. What is its job? Renal artery is a branch of this aorta. It is a branch of the aorta and it brings the oxygenated blood into the kidneys. Fine. So oxygenated blood to the kidney. Part number 4, the blue one, that is your renal vein. what does it do? It will take away the deoxygenated blood from the kidneys. So deoxygenated blood from kidneys taken away by the renal vein, and it will go and join the vena cava. Fine. Number 5, these are your ureters. What is the job of the ureter? Excuse me. The ureter carries urine from kidneys to the bladder. So from the kidney here to the bladder, which is number 6 down there. Fine. The ureter will be carrying the urine. So 6 is the sac-like structure. As you can see that it is just like a sac. So this sac-like structure has the job of temporarily storing the urine up to a certain limit. Okay. So what we say is that urinary bladder can store urine. 7 here is your sphincter muscles. What kind of muscles are these? These are circular muscles which are under your nervous control. Okay, and these will help in the release of the urine. So say at any location you feel the urge to urinate, but urine automatically doesn't pass out of your body. You don't urinate or micturate, right? Only when you're in the correct place, you have the situation to urinate, then you will urinate. So that is, the sphincter muscles are under your nervous control. And the last part here, number 8, refers to the urethra. What's its job? The urethra allows the urine to finally pass out of the body. I guess it's fine. What's this telling us? Identify the type of pressures shown in A, B and C in a plant cell, also label 1, 2, 3. So first let's see pressure A. Pressure A is your turgor pressure. That is the pressure exerted by the cell contents on the cell wall. So what is this pressure, children? It is the pressure exerted by the cell contents. Don't confuse, by cell contents on whom? On your cell wall. Fine. Look into the diagram. The opposite direction is being shown here by B. So this is your wall pressure, and wall pressure is the opposite direction. That is, wall pressure is by the cell wall, right? By the cell wall, and where it is giving to on the contents, on the cell contents. Okay, on the cell contents. Then pressure number 3, see that something is entering into the cell, right? Crossing the cell membrane, cell wall cell membrane ko cross karke something is entering into the cell. So that is the osmotic pressure which has been developed due to osmosis. Fine. A, B, C done. Now we have to label 1, 2, 3. So number 1 is your plasma membrane, right? That is the layer which molecules have to cross to enter the cell. Number 2 here, down, simple, ekdam outermost. So this is your cell wall, right? Then number 3 we have inside the cell contents. So we call this to be the vacuolar sap. Okay, the vacuolar sap, the cell contents. So I guess this one is clear for all of you. Acha, now another this experiment. So the figure shows a setup to demonstrate a certain process. Name the process. Very simple, it is osmosis, right? Now what is osmosis? Osmosis is the movement of solvent molecule, that's water, from a less concentrated solution to a more concentrated solution. Okay, have we understood this properly? Let me repeat this once. Right, osmosis is the movement of solvent molecules, water, from a less concentrated solution to a more concentrated solution. Less concentrated solution का मतलब क्या हो गया? More water, Less concentration though. That means dilute, more water. More concentrated matlab what? Less water. That is a concentrated one, not a dilute one. And what is another condition? That this movement of water occurs specifically via a semi-permeable membrane. Through a semi-permeable membrane, a good example of which is your cell membrane, selectively permeable membrane rather, and here in this case artificially semi-permeable membrane is the cellophane paper. Okay. Now what is the observation after an hour? So after an hour we are going to observe that this level of solution inside the thistle funnel increases. Now you'll ask, ma'am, why? No, nobody should ask. Y'all are ready with all the concepts by now. Just recap. Just joking. So the water is present outside, and inside the thistle funnel you have the sugar solution, right? So outside you have more water, inside you have less water. So where will water want to go? From where it is more to where it is less. So water will go inside, and as a result, if this is the level at first, now the level will go up. The level of solution inside the funnel will rise. Okay, and also you will be able to see a very less shift in the water solution, water level outside in the beaker. Say it was here, now it will fall down to this level because water has entered into the funnel. Fine. Will there be any changes? Okay, yes. Muslin cloth. So muslin cloth, how is it, like what's its permeability? Muslin cloth is a freely permeable membrane. So due to that, there's no barrier that only solvent will pass. Every molecule will pass through, right? So as a result, there will be an even level or a common level of solution in between the thistle funnel and the beaker. Okay. Rubber sheet, that is again impermeable, right? So there's no change. Neither solvent nor solute, nothing is going to pass. So, okay, so this is an important experiment for your osmosis. I guess we are clear with this. Okay, so what is the certain process? That is transpiration. What is transpiration? Transpiration is the evaporative loss of water. Evaporative means it is in the form of water vapor from the aerial parts of a plant. Sorry. Okay. So it is the evaporative loss of water in the form of water vapor from the aerial parts of a plant. Aerial means that the stems, your leaves mostly, hai na? All those. Fine. What is the paper kept? So this paper is your cobalt chloride paper. Cobalt chloride paper is originally blue in color when it is dry, right? And when there is moisture or water present, then it turns its color to pink. So that is why there is color change in B, that the plant has transpired, produced water vapor. So that water vapor has been absorbed by the cobalt chloride paper and it has become your pink color. So when dry it's blue and when it is moist it is pink in color. Okay, so this transpiration simple experiment is clear. Okay, who is this now? Identify the instrument. So this is your Ganong's potometer. Ganong's potometer. State its purpose. The purpose of this instrument is to measure the rate of transpiration, right? So its state or its purpose to measure the rate of transpiration. Fine. Now, it will measure the rate of transpiration through a cut stem. Just additional. Through a cut stem. Fine. Now, let us label the parts. Part number 1 is colored water. So you can track that how is the water flowing. Part number 2 is the air bubble. So you're basically going to track how the air bubble is moving. How far is it moving? You'll measure that in the scale. Part number 3 is the reservoir. And part number 4 is the stopcock. Right? Part number 5 is again the water. So the concept is that the amount of water that is getting transpired or that is lost by the cut stem is equal to the amount of water that this is absorbing, right? Or rather this plant is absorbing. Amount of transpiration is equal to the amount of water which is being absorbed by the plant stem. Is this clear? Okay, let's check. So what is the diagram here? The diagram is showing you a vertical section of a part of leaf where there's diffusion of water from high to low concentration. High inside to low, which is actually outside in the atmosphere. Number 1 is the waxy coating on top. So this is your cuticle. Okay. Then number 2 is the cells right on top. So that is your epidermis, and since it's on top, it's your upper epidermis. So 2 is upper epidermis. Then part 3, these tall cells that are present, these are your palisade mesophyll. Okay. Then below that, number 4, these are your spongy mesophyll. You can also call this to be palisade parenchyma, spongy parenchyma. A lot of times you all get confused that palisade comes first or spongy comes first. How to remember? P, Q, R, S. So P before S. So that is why palisade first, then spongy. Right? Part number 5 is the lower epidermis. Part number 6, these are the water vapor that is present. And then you have number 7, the guard cells. In between you have the stomata through which the water molecules are escaping outside. Okay, identify the diagram. This is a stomatal apparatus. Okay, then part number 1 refers to these one. These are your guard cells, right? The bean-shaped cells that are present, these are your guard cells. Then structure number 2 refer to the chloroplasts that are present inside the guard cells. So the guard cells inside contain the chloroplast. Then you have part number 3, which refers to the gap inside, which is the stoma, right? Part number 4 are your epidermal cells outside the guard cells. Fine. So that's it for this. I guess it's clear. Well, so that's it for this video. I hope it will be useful for your board exam preparation. Stay tuned with us. We'll be coming with more of such practice videos for y'all. If you like it, then please like, comment, share and subscribe to our channel by Explaining by Anushka. Bye-bye.